What's up, Bio Penguins? So today we're going to go and work through Unit 6, which is on gene expression and regulation. So real quick, you're probably wondering, well, why do I call y'all penguins? And why do I do this whole thing with AP Bio Penguins? And so the reason why is because penguins are in those nice little tuxedos. And so as penguins, you are now dressed for success and you are ready to rock and roll. And so welcome to being an AP Bio Penguin. So in case you don't know, which I'm assuming you all do know about these resources, there is daily review going on Instagram. At this point, I'm all the way through with Unit 6, and I start on Unit 7 on Monday. There's a 374-page review guide that's on Weebly. So if you go to my website, apbiopenguins.weebly.com, there is this huge review guide for you there. I go through the entire CED, where I have I-CAN statements, and then I break it down into topic questions. And then from those topic questions, I have also organized all of the FRQs and all of the released multiple choice questions that are in our possession at this point. Plus, I added just a couple little quick questions for you in there. I also have recorded FRQ Fridays. At the point of this video, I have done every single FRQ going from 2013 to 2023, minus 2020. And I've broken down every question. I've shown you the exemplars. And so those are all on my website ready for you to go. So you can go and practice FRQs and then watch that video, or you can pause the video, answer the question on your own, and then immediately check and see if you have that answer right. This weekend, I'm going to work through the 2 that are in the CED, and then at that point, I'll be done with all the FRQs. There's 120 quizzes games that are on my website. So again, those are all open and available for you to play at any point. And then there's review PowerPoint. So honestly, click around the website, you will find tons of resources, and they are all 100% free for you. So the plan today, we're going to go through molecular genetics. We'll talk about operons and we'll talk about biotechnology. I have a couple practice questions. I think I have 3 multiple choice questions and 2 free responses in here. And then I'll open up for any type of questions and answer. Okay. As I work through this, I find that there's like a slight lag. And so if you have a question, you're welcome to throw it into the chat over here. But I'm going to kind of keep working through my presentation. But if I can answer your question, I'll jump back and do it. In previous sessions, I've seen the students will answer each other in that chat. And so if you have an answer to what someone else is saying, then you're welcome to answer that question for them. So let's get jumping into this, right? So central dogma. So the central dogma states that we go from DNA to RNA to a polypeptide. And so the genetic material being DNA. And so if we go through replication, okay, replication is where we have DNA and we use that DNA to make a copy of itself. You can then go from DNA to RNA. That's the process of transcription. Okay. Then if you go from RNA to a polypeptide, we'll say that's translation. The reason we don't say this is a protein is because there are some times where our proteins are made up of multiple polypeptides. So the process of the central dogma is making the polypeptide, not really making the protein. So examples of different proteins that may have more than one polypeptide. So like hemoglobin is made up of 4 subunits. Collagen is made up of 3. And so most of our proteins actually have multiple polypeptides that work together. So if you remember back to Unit 1, the levels of folding, that quaternary structure was when we had more than one polypeptide that was bonded together through those R groups. So now it is important that we understand that there are some things that violate that central dogma. So one of those would be our retroviruses. Our retroviruses are going to have an RNA genome. And so from that RNA genome, they have an enzyme called reverse transcriptase. Well, at least HIV does. And that allows it to go through reverse transcription. And it can take its RNA genome and then make it into DNA. So it uses the RNA as a template. It base pairs up to make a RNA DNA kind of hybrid. And then it uses that DNA to make a second DNA strand, which then it will allow itself to insert into the host DNA. And that's how HIV actually works, is that it inserts itself into your white blood cells. And every time you make copies of your white blood cells, you make copies of the virus until it then, of course, exits and makes this little viral, all that fun stuff. And so in terms of replication, we have a couple big things to understand. Number one, where it's taking place is different between a eukaryote and a prokaryote. Okay, remember, eukaryotes have a nucleus, prokaryotes do not have a nucleus. So in terms of eukaryotes, we'll see replication taking place in that nucleus, versus in a prokaryote, it will take place in that region, which is called that nucleoid. Okay, the structure of DNA is slightly different. So eukaryotes have multiple strands and it's linear. So it's just these long strings of DNA. versus a prokaryote has a singular circular piece of DNA. And when I say single, I don't mean that it only has one strand. It's still double-stranded because by definition, DNA is double-stranded. Yes, I do know there are some times and some violations of that where sometimes DNA is single-stranded, but for the most part, DNA is double-stranded. So when I say single circular, I mean that they have one strand and they have one DNA double-stranded and it's just one circular. They have plasmids that we've talked about in other times, but when we talk about its genome, it's literally a single circular genome. And so different other reminders that we have. So like DNA is made up of adenine, thymine, cytosine, and guanine. Okay, it has a pentose sugar of deoxyribose, and then it also has a phosphate group. So those three things together would be your nucleotide. And if we didn't have the phosphate, it's a nucleoside. That's why we call it adenosine triphosphate, is because of the fact that it's the nucleoside of adenine, and then actually ATP is looking at ribose, but whatever. Okay. And then also when we're looking at DNA, we have the purines, which would be our double-stranded ones. So adenine and guanine. So you can think about like pure silver AG. And then your pyrimidines are going to be your cytosine and your thymine. Okay. And so when we kind of think about all of them, you remember cut the pyramid, so CUT, but since DNA doesn't have uracil, we just say C in the T. And so it's important that we understand that purines have a double ring, pyrimidines have a single ring. So I tell my students that a big name means that we have a small structure, so a single ring, versus a small name of purine has a double ring, so that big structure. And then it's important to know the double bonds. And I actually was discussing this with another teacher, like the students don't need to know that. Well, yeah, you do. It's directly in the CED, the curriculum that we follow, that you have to know the number of bonds. So A and T has 2 bonds. C and G has 3 bonds. And this is the hydrogen bonds. And so the way that I can remember that is that it takes basically two lines to make capital T. So 2 for adenine and thymine. And then cytosine is the third letter in the alphabet, so 3 for the C. And so if you can remember that A and T go together and that C and G go together, you'd be good. So ways that students can remember that is that apples are in the tree, so A and T, and then the cars go in the garage. And then when we get into RNA, we talk about uracil, you could talk about that the apples are under the tree. And so there's the A and the U could be together. And then when we think about the structure, we've got a sideness to it. Okay. At your 5' end, there's a phosphate. You don't really need to know the counting of it, but in case you really, really wanted to know, the way that we count around the structure, we've got 1, 2, 3, 4, 5, and you're counting the carbons on it. And so on the first carbon, we see that you've got your nitrogenous base. On the third carbon, where the hydroxyl group is of that sugar. And then on that 5th carbon is where we see the phosphate. So that's why we say the 5' end has the phosphate and the 3' end has the hydroxyl group, and that they're going to be anti-parallel. Okay, and so where I have a 3' across from it's going to be a 5', and when there's a 5' across it will be a 3'. And that's going to be important when we talk about the way that replication takes place, and being able to eliminate some of our answer choices. Okay, also directionality is important. DNA is read 3' to 5'. DNA is made 5' to 3'. Again, anti-parallel. Okay, so students trip that up all the time. So it's important that we remember that we read 3 to 5 and we make 5 to 3. Again, anti-parallel. And so in terms of replication, there's a couple different steps. And I attempted to make an animation. So here I have helicase. Now I know I already separated them, but it's okay. So helicase is going to go through and it's going to unwind the DNA strand. The way that it unwinds it is that it's going to break the hydrogen bonds that we have in between those nitrogen spaces. As we said before, adenine and thymine have 2, cytosine and guanine have 3. The way that they could kind of expand this and ask you questions, they might ask you about like the melting point. So if there's more adenine and thymines in a structure, a DNA structure, it's going to have a lower melting point than if it has more cytosines and guanines. Okay. So after the DNA, of course, pulls it apart. The helicase breaks those hydrogen bonds and pulls them apart. And of course, it's not taking the whole strand apart, it's only taking a little section. Top isomerase is going to relax that supercoil ahead of the replication fork. When I say replication fork, I'm just talking about where is the point where they're coming apart, okay? Because it's going to make like a fork in the road almost. And so these two strands are together, it opens it up and makes that fork that you might see in between there, okay? So now, way upstream, if you think about any time that you take two things that are coiled together and you pull them apart, upstream is going to get supercoiled. And so that's the job of top isomerase, is that it's going to break the double bond, unravel a little bit, and then it's going to put those back together, that phosphodiester linkage, okay? So then we're going to have primase, okay? So DNA polymerase is unable to start from scratch. So it's going to need a primase. Primase's job is to make a RNA primer. So what's going to go through is it's going to read, again, 3 to 5. It's going to synthesize 5 to 3. So what does this look like? So what we're going to see is we're going to go through and have the base pair, right? So cytosine and guanine will pair together. And again, you see cytosine and guanine pairing together. So the primase is going to go through and it's going to put on that primer. Okay, and so it base pairs up, adenine and thymine, cytosine, guanine, and it does the same thing here. Now, again, since this is RNA, we're going to have uracil in there instead of thymine, okay? But that will get replaced later on with DNA polymerase will come back in and fix that later, okay? And then the next thing will happen, since I've got my primer, now DNA polymerase can come on, okay? And so I just finish this out by having the helicase open up the next part, and then polymerase continues on. So DNA polymerase's job is to synthesize the DNA polymer. Enzymes tell you what they do. Helicase breaks apart the helix. Primase makes a primer. DNA polymerase makes a DNA polymer. Top isomerase, I guess you could think about like a spinning of a top, but it's going to unwind the part, okay? And so we have these two different strands. We've got our leading strand and we have our lagging strand. Our leading strand is going to be synthesized continuously toward the replication fork. So as I said before, we're going to have this replication fork, okay? It's going to be where helicase is pulled apart the two strands, okay? So the one that's moving towards that replication fork, okay? So my upper strand, the one that was 3 to 5, is being synthesized 5 to 3 going toward that fork. Now our bottom strand is going to have a 3' end here and the 5' end there. And so it's going to synthesize the opposite direction. That is going to be my lagging strand, okay? And so it's going to move a little bit, make the little strand, and then open up a little bit more and makes the next strand. And so we're going to see this going to have to keep doubling back and synthesizing that strand. It's going to keep kind of jumping, if you may. And that's our lagging strand. So since I have these little fragments, the first fragment here, the second fragment here, or in this case on my diagram, the first fragment and the second fragment, I need ligase to come in. So ligase is going to come in and is going to seal that bond for me. Okay, so then I can make my full strand. So now I see a question came in the chat. Why are there different melting points depending on which one's binding? It has to do with the number of bonds. So adenine and thymine have 2 hydrogen bonds. And so since if I would talk about a DNA strand that has multiple adenines and thymines in it, that means there's a whole bunch of just 2 hydrogen bonds in there. And then if I was talking about a different strand that had more cytosines and guanines that have 3 bonds between those 3 hydrogen bonds, then that's going to be more hydrogen bonds, which is going to allow it to have a higher melting point. It requires more energy to break all those hydrogen bonds than it does with the lower one. Okay, and it does look like that somebody answered it. Thank you, Nisha. I hope I said that right for answering that question. So moving into transcription, okay? As we said before, transcription is where we're going to read the DNA and we'll synthesize the RNA strand, okay? So again, location is important. Eukaryotes will have this take place in our nucleus, and the prokaryotes will take place in the nucleoid. Keeping in mind that is in the cytosol, right? There's no membrane that separates it in a prokaryote, okay? Reminders about RNA. Ooh, that's a typo. That should say RNA. I'm so sorry, y'all. RNA is made up of adenine, uracil, cytosine, and guanine. It has the pentose sugar of ribose. So that is different than DNA. So if we're thinking about the differences between DNA and RNA, DNA had a thymine, RNA has a uracil, DNA had deoxyribose, RNA has ribose. So those are big differences we see there, okay? So then purine again is adenines and guanines, pyrimidines is cytosine and uracil. And so now that I have them all, kind of think about that you would cut the pyramid, C-U-T, to help you remember the pyrimidines. And I see the question, why can't it go 3' to 5'? This does sound dumb. It is, why can't? Okay, so RNA polymerase is an enzyme, and enzymes have active sites. And so my only assumption would be that in order for the binding of the new strand, it would have to bind in a certain way, and that way happens to make it 5 to 3 instead of making it 3 to 5. Okay, so excuse me. Additionally, we think of our base pairing rules. So adenine and thymine if we're thinking about DNA, but it's adenine and uracil when we're looking at RNA. Again, 2 hydrogen bonds, cytosine and guanine with 3 hydrogen bonds. There is the same sideness where we have a 5' end and a 3' end. In terms of our 5' is our phosphate, 3' is our hydroxyl, and the directionality is the exact same as the DNA. So the way that I explain this to my students is that it's the same language. So if you were to right now write down everything I say, I'm speaking in English to you, okay? And you're going to be writing it down in English, okay? You're transcribing it in the same language, okay? So that is the way I explain it. So you're going to read the same direction, you're going to write the same direction. It's the way that English works. It's, of course, read one way, yeah, okay? And so we need to make sure that we're looking at our template strand, Okay, so there are multiple names that this can be called. And in the CED, all 4 names are in there, which means that these can be used interchangeably in a question. Okay, they can call this the template strand, they can call it the non-coding strand, the minus strand, or the antisense strand. All 4 of those terms can be used to describe the template strand. The template strand is the one that is 3' to 5' that is being read on DNA to synthesize the RNA. And when I say that it's being read, it's the one that's going to be base pairing up to make that RNA strand. Okay, so here again, we've got my transcription. RNA polymerase is going to be able to separate them. And I don't know if you caught that. The enzyme we're using here is RNA polymerase. We're making it RNA polymer, so we're using RNA polymerase. Okay, so it's going to be able to pull them apart. It doesn't need any helicase. The RNA polymerase is able to separate them in that spot. Okay, so RNA polymerase is going to synthesize the mRNA strand in the 5' to 3' direction, reading that template DNA in a 3 to 5, as I've already said. So RNA polymerase comes through and it base pairs it up. Yes, I'm so excited about this animation. Like, I'm proud of myself for this animation. I worked really hard on this. So base pairs up. Anytime that I have an adenine, there's going to be a uracil, but other than that, it looks the exact same. So a time saver. Okay, so if you're trying to work through something on the exam and you're like freaking out because you have to a time crunch, okay, keep in mind that your template strand, okay, sorry, I said that wrong. The one that you're going to base pair, the mRNA that you're making, looks just like the one that's not the template strand, the non-template strand. Okay, looks the exact same. The only difference is that there's a U. So if you're needing to quickly look at it and you're needing to quickly make that mRNA strand, just look at the 5' to 3' strand and just switch out all the T's for U's, and you got a new strand. You got the right one. Okay, so when we think about this, there's a promoter. This is the site where RNA polymerase is going to bind to start transcription. Okay, and then there's going to be these things called transcription factors, which are your activators or your inhibitors that will turn on and turn off gene expression. So when we looked at cell communication, we talked about this transcription factor. We mentioned how it can go into the nucleus and it can turn on a gene. Well, the way that it does that is it's going to bind to a site, which is then going to allow for the RNA polymerase to bind. So I always kind of think about it like a baseball glove. If you're trying to catch a ball, you don't want to catch the baseball barehanded. You're going to want there to be a little bit of extra support. And so the activators are going to kind of act as that, right? Our transcription factors are going to extend the region, and it's going to draw the RNA polymerase to that site, and it gives it nice secure binding so it can then read that DNA to synthesize that RNA strand, okay? So after we go through this, after we've made our RNA, well, there's slight differences between a prokaryote and a eukaryote. A prokaryote doesn't have a nucleus, so it's going to immediately start translating. As soon as we start transcribing it, it's going to start translating it. Okay. Versus in a eukaryote, we can do some stuff to that RNA before we allow it to leave the nucleus. Real quick, there was a question: what's the difference between a promoter and a primer? The promoter is a site on the DNA. It's just like a certain coding region, and that's where the RNA polymerase is going to bind. Versus a primer is just talking about, I guess, it's something that's going to start something. So like a primer will be like the initial like point. And so like when we're looking at the DNA polymerase, we make a strand that's like a primer. So like crystals, I don't know if you've ever noticed like how a crystal is formed. They put like a piece of sand in there first, and then that sand gets built upon. So our primer is what's going to start the synthesis of that DNA strand. It's giving something to build off of, versus the promoter is just like a region. So I hope that I've explained that for you. Okay. Yeah, mutations later on in this. And so there's 3 different things that we're going to see is going to take place with our post-transcriptional modifications. Again, this is taking place only in eukaryotes because of the fact that transcription and translation can take place in different regions, right? Transcription will take place in our nucleus, versus translation will take place in our cytosol for both. Well, I guess in eukaryotes. So first thing's going to happen is we're going to add a 5' guanine cap. Okay, this signals the start of the transcript. Okay, this is where the ribosome is going to bind, is at that 5' end. So I need to put a guanine over here to say, hey, this is the start of it. This also facilitates the export from the nucleus. So this is what's going to allow the RNA to leave the nucleus and come out into the cytosol where those ribosomes are. We're also going to remove introns. These introns are going to be intervening sequences. They're non-coding regions. There's just junk in there. So this is like if you had a textbook or any type of book, and there was like chapters 3 and 4 are just blank pages. That's a waste of resources to have to sit there and flip through every single page of your book. So what we're doing is, after we've realized, hey, there's this part that is blank in here, we're going to cut that part out so that you only have the coding region. You only have the words in your textbook. And so we're going to cut that out. And that's where we get the alternative gene splicing, is that we can cut out different introns and exons during that kind of splicing point. And then we're going to have a poly A tail. The poly A tail is just a whole bunch of adenines that we put at the end of the mRNA. And it's designed to decrease the hydrolytic enzymes. There's these enzymes that are in our cytosol, and their job is just to basically cut from that the 3' end, and it makes our strand smaller. And so as we're cutting like the functional parts of that protein or the polypeptide, then we're not going to be able to make the correct protein anymore, because we're cutting out that coding sequence that made that polypeptide. And so this kind of prolongs the life of the mRNA. So 3 things that happened: we had a 5' cap, we spliced out the introns, and we had a poly A tail. So in terms of translation was that last step when we make the RNA, and we're now going to read that RNA, and we're going to synthesize a polypeptide. So in prokaryotes, they happen simultaneously, while eukaryotes, they add the cap. And yes, so in terms of prokaryotes versus eukaryotes, prokaryotes can, as soon as they are done scribing, like, so they like literally make the first little blip of the RNA, the ribosome immediately will attach to it and it can start translating it. And so there was a question a couple of years ago, an FRQ question, where they asked the difference between a prokaryote and a eukaryote in terms of the length of a strand. And you would say the strand was the exact same length because it didn't cut out the introns, versus a eukaryote did cut out those introns. I think that may have been 2018, but I could be wrong about what year it was. Okay, so in translation, this is our ribosome. Our ribosome is made up of our RNA, and then it's made up of proteins. There's 2 parts to it. We have a large subunit, and we have a small subunit. The large subunit is going to bind to my tRNA, my transfer RNA that is transferring the amino acids to that ribosome. And then the small subunit is binding to the mRNA. So this is actually where the 3 different types of RNA that we think about and we talk about are all working together. So our RNA makes up the ribosome, our tRNA is going to bring the amino acids to the ribosome, and then the mRNA has the message from the DNA. And so locations, as we said, eukaryotes, this will take place in the cytosol or the rough ER, and in prokaryotes, it takes place in the cytosol. And so just real quick, in case you don't know, all ribosomes start out being in the cytosol. They'll get a message that allows them to move to that rough ER. So we will see ribosomes on the rough ER, but they do all start as cytosolic ribosomes. And so in terms of our translation, there are 3 steps. We have initiation, elongation, and termination. Initiation is going to start at AUG. I don't know where you are, but for me, school starts in August, AUG. And so that kind of helps me remember the start codon is AUG. You do not have to have them memorized. They're going to give you this beautiful chart on your exam. And so if you look right here, where is it at? MET. AUG, we have MET, which stands for methionine, which is an amino acid, but it also stands for the start. So the very first amino acid that you're going to have in every single polypeptide is going to be a methionine. I did learn a couple of years ago when I was doing one of the practice questions, they actually do cut that methionine off later, but in terms of when you're working on the exam, AUG is going to be your start, and it codes for methionine. Elongation is where we're going to base pair between the tRNA and the mRNA to add each of the amino acids in. And don't worry, I made an animation for it. I'm super excited about it. And then termination is when we reach a stop codon. So we'll have UGA. So if you are a UGA fan, University of Georgia, you'll know that they are unstoppable. Whatever. There's also UAA and UAG. Again, you don't have to have these memorized because they are given to you on the codon chart. Okay. And so in terms of translation, there are many steps. We said we are going to have start codon, right? So I already am going to assume that we're already started on this, that we already had the AUG come in. And so we're going to talk about how does translation take place, right? So there's 3 sites. You have the A site, oh my gosh, the A site, the P site, and the E site. Okay, so the A site is where we're going to add the amino acid in. Okay, so we're going to have a tRNA that has the amino acid and it's going to come in. The anticodon, which is going to be on our tRNA, is going to base pair up with the codon that's on my mRNA. So here we see C, U, and then C, because it will base pair up. So if you're trying to figure out where is that on the chart, okay, you're going to look on your codon. So GAG is right here. So that's going to tell me that I have glutamate. And so my glutamate is going to be attached to that amino acid. In case you're trying to figure out how to use this codon chart, your first base is going to be over here. So this is my first one was G. I look down here, I find the G. So I know I'm in this kind of row right here. My second one was an A, so I know that I'm in this box right here. And then I look for GAG, which is right here, so glutamate. And you can use, again, this chart given to you on your exam, I promise. Okay, so we We have our anticodon pairing up with our codon. So then what's in the A site shifts over. We translocate. Okay, so we shift over from the A site to the P site. And so this is where we're going to have our growing polypeptide. Now the A site's open and another amino acid is going to come in, right? So the tRNA, again, base pairing up. I guess I didn't put the animation on there to base pair up, but so we base pair up and we have our serine. So we see UCG, makes serine, and then of course we translocate. And when we translocate, now we have the thing in the E site. So my E site is going to be my empty tRNA ready to exit, okay? And so we see the glutamate and the serine is going to now be bonded together on that P site. Okay, we have our polypeptide. So after that, we then shift over again. We have another one base pairing, and it's going to keep on going through that shifting until we reach the stop codon. So if you see right here, I've got, where am I at? I guess I just made a stop codon just to show you a stop codon. And so at that point, a water molecule will come in, which will allow our polypeptide to come apart. The whole structure will fall apart. And then we, of course, have our little polypeptide. Now, of course, it would be much longer than that, but you get the idea. What are the proofreading mechanisms in DNA replication? In terms of proofreading, we just see that DNA polymerase is going to be able to kind of check itself. And so we'll see that just like as you're typing along and you make an error, if it realizes that it made the error, it just backspaces a second, fixes it, and then goes on. DNA polymerase can also check the DNA strand, and it notices if there's a mismatch, in which it will then use a nuclease to cut out that segment, and then DNA polymerase will come in and fix that base pairing rules, and then we'll see again ligase will seal that back together. So you'll kind of see that that will take place, and you don't need to know the differences between the types of DNA polymerase. You just have to know that DNA polymerase does it. So DNA polymerase changes out the RNA primer back into DNA primer. It synthesizes the DNA polymer. It is going to also check for the different errors. When do mutations occur? We're going to talk again about mutations. We have not talked about mutations yet. I explained a little bit about termination part. And so there isn't a tRNA that's going to come in and bind because of the fact that this is the stop codon. Instead, a release factor comes in. And so when that release factor comes in, that signals for a water molecule to come in. And so we know that when you use water, we go through hydrolysis. Hydrolysis will then break that bond. So that water molecule will break the bond that's right here between the tRNA and that polypeptide. And so it'll break that bond using that water molecule, which then allows the whole thing to fall apart. DNA polymerase 1 or 2 are all DNA polymerases. We don't even know the differences between the two, so I'm going to be honest with you, I don't have them memorized because I haven't really needed to memorize them, and since before the 2012 curriculum is when I needed to have those known. Okay, so mutations. Point mutations, mutation at one nuclease base. Okay, so when we think about point mutation, we have a mutation in a single point, 1 point. Okay. So we can have a silent mutation. There's no change in the amino acid. AA stands for amino acid, in case you're wondering, okay? So UUA and CUA are going to code for the exact same amino acid. So although there's an error, although it's a different in that nucleotide, we still code for the exact same amino acid. So you don't notice an error, so we call that a silent mutation. Versus a missense mutation, I changed from one amino acid to another amino acid. So it's like if I, instead of talking about I wanted to walk my dog, I say that I walked my fish. Well, that doesn't really make sense in that sentence, right? That's missense. Instead of a dog, it's a fish. So you're just like, but it's just a changing of one amino acid. There's which one amino acid that changes. Versus a nonsense is going to be where we have a premature stop, okay? So I'm telling you a story. I tell you all about how my son jumped off the bed and then we had to take him. And I just stopped. Like, you don't know where the end of the polypeptide is. You don't know where the end of the sentence was going because I just stopped. So it's like a premature stop, okay? So silent has no change. Missense has changed from one amino acid to another. And then nonsense is just completely to a stop codon. Okay, versus a frame shift mutation deals with that there is an insertion or a deletion of one or two nucleotides, and that causes you to shift the reading frame. So instead of saying the cat ran too far, I say takaufa. Well, what? Takaufa? That doesn't make any sense. But what is happening is that I've shifted my reading frame. So instead of reading the cat ran too far, I shifted by one letter, making it takaufa, which hopefully doesn't say anything bad, but whatever. Okay. So if we're talking about an insertion or deletion of 3 nucleotides, it's not going to do that because our codons are in groups of 3. And so I may add one amino acid, I may delete one amino acid, but it's not going to shift all of those groups of 3. Okay. I was going to say something. Okay. So when we get farther and we're talking about like cladograms and we're talking about unit 7 and talking about differences, the silent mutations, the reason why we're going to say that DNA is more accurate to use than amino acids, because DNA is going to show all those changes versus if I just look at the amino acid, I wouldn't pick up these silent mutations because they're the exact same amino acid. So we can also have chromosomal mutation. This has to do with where we rearrange a certain part or change that chromosome number. So that's where we have insertions, deletions, duplications, inversions, or translocations. Insertions where we can insert something, deletions where we delete something. Duplication, we make multiple copies. That's what's happening with like Huntington's disease, that they duplicated that gene too many times. Inversion, it comes out, it flips over and then pops it back in. And then translocation, it just moves it to another chromosome, which is where Down syndrome sometimes comes from, is that there is a kind of a translocation where it takes a portion of chromosome 21 and puts it on another chromosome. I can't remember which one it is. Maybe 13, maybe 15. It doesn't matter. And then we can also have changes in our chromosome number. So like non-disjunction, we talked about, I believe, in Unit 5. Where we saw that you could have like 2 of our chromosomes do not segregate during anaphase, which leads you to having an additional chromosome in one of your gametes and not in the other, and be missing a chromosome in one of your gametes. And then polyploidy means that all of the chromosomes, there's a whole nother set. So like a triploid has 3 sets of chromosomes, a diploid has 2 sets of chromosomes, a tetraploid has 4 sets of chromosomes. So it's just being, you have multiple sets. Okay, what happens if the tRNA binded is the wrong amino acid for some reason it goes in? So because of the fact that we have that base pairing rules, you're not going to put the wrong amino acid. So there's a specific tRNA aminoacetylase, which is the enzyme that is going to add the tRNA and the amino acid together. So there's that specific binding to that active site. So you have the correct amino acid put on the correct tRNA. And then you're going to see that that tRNA is going to base pair up with our mRNA. So we have the correct basing there to bring that amino acid in. If there is an error, it's probably just going to take place at one location. You made that one misfunctional protein. It's not going to have a problem downstream because of the fact that it was this one time. Versus errors that we see in our DNA. This error right here that we're seeing, these were errors that took place in our DNA. And so every time I transcribe and translate that gene, I make this same error. This same problem is taking place in that. And so that's why we see that this is a recurring problem. So like thinking about any of those not the chromosomal, but like point mutation diseases, like sickle cell or Tay-Sachs or one of those, it's in the DNA. So every protein is formed non-functionally. So in terms of operons, okay, so operons we only find in prokaryotes, okay, so this is the gene regulation we see it's, because it's very simplified. So we're going to have, again, the promoter region, which is where our RNA polymerase is going to bind. We have our operator. This is the site where a repressor binds. And then, of course, we have the genes. So there's those 3 things that make up these operons: promoter, operator, and genes, okay. So I like to think of this like a railroad track. Okay, so the promoter region is where we're going to have our train, being our RNA polymerase, is going to bind. Okay, the operator is going to be a specific site on that train track. Okay, and then of course the train tracks is the DNA. And so a repressor would be like a big rock. So if my big rock is on top of my train tracks, can my train get on the track? Well, no, it can't. And so this is going to inhibit that train from moving on those tracks. It's going to inhibit RNA polymerase from binding, and it's going to inhibit it from being able to transcribe that gene. So we have 2 different types of, we have repressible operons and we have inducible operons. So let's first think about repressible operons. The job of a repressible operon is that it's going to be on and it's going to get turned off. Okay, and I know I say on and off 20 million times with this. I don't really know any other way to say it. So it's going to be on and off for a while. And so with this, an example of that would be our trip operon. I don't know if they'll use trip operon on your exam, but it's the main example that we usually use. The job of it is going to synthesize tryptophan. So we find that most of our repressible operons are going to synthesize something, okay? There's some type of anabolic pathway. They're going to synthesize some big thing, okay? And so we're synthesizing tryptophan. Well, the bacteria, E. coli, lives in your intestines, okay? And so when you consume something, when you eat food, right, your body breaks down that food and you're releasing out these amino acids, specifically tryptophan. So if there is tryptophan in your diet and you consume the tryptophan, does the E. coli need to make that tryptophan? Well, no, it doesn't. And so that's why we're going to call this a repressible operon, because if tryptophan is present, it's going to turn that operon off. Well, why do I need to make something if you already gave it to me, being the E. coli? Okay, so it starts out being on, and the repressor is inactive. Okay, so the repressor is not bound to the operator, and so we're going to be synthesizing that tryptophan. If that trip is present, tryptophan is going to bind to the repressor. So if tryptophan binds to the repressor, okay, it's going to activate my repressor. My big rock is now there. And the repressor binds to the operator. So if the repressor is bound to the operator, I cannot have RNA polymerase binding, and so my operon is now off. Since TRIP is there, TRIP binds to the repressor, right? Making it so that I'm not going to make any more TRIP. I don't need to make something I already have. Versus an inducible operon, its job is going to be to turn something on. We're going to turn the gene on with this. So an example we have is the lac operon. These would be our catabolic pathways, okay? I'm synthesizing something to break something else down, okay? So it starts out being off, and the repressor is active. So that means that my repressor is always bound to my operators, always there, okay? And so if lactose is present, lactose is going to bind to my repressor to inactivate it. It makes it so that it's no longer bound. And since it's no longer bound, RNA polymerase can bind, and it synthesizes the enzymes to break down lactose. Why do I have to break down lactose if lactose isn't there? So it waits until the lactose is there before it can do it. Operons are definitely a negative feedback loop. Both of these are negative feedback loops. There's a specific type of positive feedback that takes place with one of them. It's CAP, I believe. There's like something that works with this, that if there is a high amount of lactose and a low amount of glucose, I think, it's going to try to benefit on the lactose versus the glucose. I truthfully cannot remember that one off the top of my head, though. I'm sorry. Okay, so these, again, I don't know which one they're going to give you on the exam. I don't know if they'll even give you lac or trip on the exam. They may kind of trip you up by giving you something different, but you just need to make sure that you're following the path. And so whenever I have these, I just kind of draw myself a picture to kind of think through it. Okay, repressible, again, think about the word repress, turn something off. So that means this one will be on and get turned off. Inducible, if you're inducing someone for labor, you're trying to get them to have the baby. So we're going to turn the gene on, right? So it turns off, it's off and gets turned on, okay? So kind of think about these as opposites. So biotech is the last thing we have in this one, right? So there's 4 different things that are in the standard. It's 6.8 in terms of the curriculum. So our first one is gel electrophoresis. They haven't released an exam in a couple of years, but every time that I've seen one, they always talk about, there's always a gel electrophoresis on there. Okay, so what we do with the gel electrophoresis is you put DNA samples into these little wells. So there's these little kind of pockets up here, the wells. And notice that this is a negative side and this is the positive side. DNA is negatively charged. If you remember that there are all these phosphate groups in it, phosphate is negatively charged. Yeah, so you do kind of need to know about functional groups in terms of like remembering that kind of stuff. So phosphate is negatively charged, making DNA negatively charged. So it's going to be kind of drawn to the positive end. And so we're going to see that when we turn the electricity on, that we're going to be able to separate the DNA based on charge and size. And so the longer fragments will stay up here by the well, and the smaller fragments are going to go down here at the bottom, okay? So it's kind of like thinking about a chain link fence. Who's going to be able to get through that chain link fence competing between you and like a rabbit? Well, a rabbit's going to be able to go in and out of the holes in the chain link fence versus you kind of get stuck at that fence. And so it's kind of like that, where the gel electrophoresis, the agarose gel, has all these little pores in it. And so bigger fragments can't go as far. They kind of get stuck versus smaller fragments can kind of weave in and out. And so what they may ask you to do is compare those banding fragments and kind of determine somebody's trait. So like they'll give you someone that's homozygous dominant, someone's homozygous recessive, someone's heterozygous, and you'll be able to figure out the genotypes based on that. I don't think they've ever asked like a paternity question, but they could ask them to have a paternity question because of the fact that half of your genetic material is going to come from each parent. So they could ask that kind of thing. So you'll just have to be able to read the diagram or be able to kind of expand on that. Another thing you might see is that the thickness of the band. So if there's a thicker band, it means that there's more DNA there versus a thinner band, there's less DNA. We also have PCR. PCR is going to be polymerase chain reaction. So thinking about chain reaction is going to happen one after the other. And so we're going to try to make multiple copies of DNA fragments. And so it goes through these 3 steps of heating, cooling, and annealing. Okay. So what happens is we heat it up By heating it up, it denatures the DNA. It pulls them apart into single strands. Cooling it down allows for the primers that we've added to the structure, okay, to bind to the specific region we want them to bind to. And then the annealing is where the DNA polymerase is going to bind and go through it. Because of the fact that we keep heating it up and cooling it down, heating up and cooling up, because it's going to keep taking place over and over inside of a thermocycler, we're going to have to use a heat-resistant polymerase. The heat-resistant polymerase is Taq polymerase. It comes from a thermophile, so a bacteria that actually lives in very warm conditions. It's going to then be able to connect that. Somebody says, can you explain how the length of DNA strand is related to the positive end? It's not that the length is related to the positive end, it's that the negative charge of the DNA is drawn toward the positive end. And so we're going to see that these smaller fragments will move farther because they are able to get pulled and they can go in and out of the pores that are in the agarose gel versus the longer fragments. They're pulled toward it, yes, but they get stuck because they can't get in and out of the pores as fast. Okay, so PCR is going to allow us to make multiple copies all at once. And so if you're trying to amplify your DNA, you're trying to make multiple copies, that's what you would go through, PCR, and then usually after PCR, we go through the gel. And then we also have a transformation. You probably did this lab with your class in which you're going to go and you put DNA into a solution with your bacteria. You then heat shock it. So you heat it up a little bit, but then you cool it right back down. And so that allows basically the membrane to open up a little bit. We're just looking at phospholipids. They kind of separate a little bit. And then this little plasmid can come in. And so then you're going to grow it onto a plate, and you're going to try to see which of them kind of grow on that plate. And so when we look at this, we're kind of seeing that we're doing kind of selection for them, right? We're doing where if there is antibiotic resistant in that plasmid, they're going to grow in the presence of that antibiotic. And so I've seen questions where like they put insulin and they put the antibiotic resistance on that plasmid. And then they ask like, what's the percent that are going to have the highest amount of insulin abilities? Well, it's going to have to be the one that has antibiotic in the presence because that's going to be the highest percent. Yeah, both of them that are transformed are going to be able to grow, but the one that's only the transformed bacteria will have the highest percent. So you have to really read closely into those questions. And then last but not least, DNA sequencing. I don't really know how they're going to use this on the exam. This is just looking at coding each of the different nucleotides so you can actually determine the sequence of those strands. Now, somebody in the chat asked, why don't eukaryotics have an operon? It's just because we're a little bit more complicated. I think I saw today in Reddit, somebody said that eukaryotes do have operons. It's not tested on the curriculum, but I've never come across a eukaryotic example with an operon. There may still be them. But the reasoning why is because our genes are just separated because we independently regulate them all. So each of our genes has their own promoter. So we would then regulate the multiple genes. So like in a prokaryote, let's talk about like, I don't know, the trip operon. Trip operon has, I think, 3 different genes that it's controlling. And so we synthesize those 3 genes all at once in a prokaryote. Versus like in a eukaryote, there'd be a promoter in the first gene, a promoter in the second gene, and then another chromosome, promoter and a third gene. And so you would have them on 3 different chromosomes, and so we just can regulate them differently. Anyway, so let's look at some multiple choice. I realize I'm running short on time. That means I'm probably not going to hit my 1 hour mark, but I'm going to do my best. So in terms of this, DNA replicates. Each strand of the original DNA molecule is used as a template for the synthesis of a second complementary strand. Which following features most accurately illustrates enzyme-mediated synthesis of a new DNA replication fork? So if you think to yourself, okay, which way do I read the DNA and which way do I synthesize the DNA? Well, DNA is read 3 to 5 and it's synthesized 5 to 3. Okay, so let's look at our first example. My first example, this one is reading 5 to 3. That's not right. So then this one, okay, we're reading it 5 to 3 and we're synthesizing it 3 to 5. That's not right. Here, let's see. So I'm, again, reading 5 to 3 and synthesizing 3 to 5. That's not right. But here, look. I read it, here's a 5 to 3, and here's synthesize the opposite direction. Okay, so what we're seeing is that I'm reading this bottom strand, 3 to 5, and making it 5 to 3. Here I see that I'm reading 3 to 5 and synthesizing 5 to 3, 5 to 3, 5 to 3. That's my answer. And so you really need to kind of look at it and figure out which of the directionality. In addition, you can kind of see that this one was anti-parallel. So this one wouldn't be showing anti-parallel. This one wouldn't show me be anti-parallel. So this one and this one both showed anti-parallel, but you just weren't looking at the strand moving in the right direction. So you kind of really have to look at it. So sickle cell anemia results in a point mutation in the HBB gene. Mutation results in a replacement of amino acid as a hydrophilic R group amino acid that has a hydrophobic R group on the exterior. So we substituted hydrophilic, which is water loving, for hydrophobic, which is water hating. And that's going to cause the shape of my polypeptide to change. Okay. So this mutation most likely will result in altered what? And so we're trying to figure out, well, why would this mutation lead to a difference? And it has to do with the fact that we're not seeing the same bonding. Okay, so properties of the molecule is a result of abnormal interaction between adjacent molecules. So that's talking about quaternary structure. I have different R groups, so different binding. That one sounds plausible. Let's keep looking. DNA structure as a result of abnormal hydrogen bonding between nitrogenous bases. This time about point mutation, and we're looking at a protein and amino acid. That's not talking about the DNA structure, so that one's out. C talks about fatty acid structure and ionic components between fatty acid chains. Again, I'm looking at amino acids, which is part of protein, so C is also not right. Protein secondary structure as a result of abnormal hydrophobic interaction between R groups and the backbone of the protein. Wait, that sounds really good. I like that one. But that one's not right. And the reasoning why is because they're tripping you up in a way. Because if you remember, the secondary structure was on hydrogen bonding. It was on the alpha helix and the beta pleated sheet. The tertiary structure is the R groups. So here on D, they make it sound really good. But that's not the answer, because secondary structure is our hydrogen bonding with our backbone. And then the R groups is part of our tertiary structure. So don't fall in those traps. If you find something wrong about an answer choice, don't pick it, okay? And then here we see operons. Lactose digestion in E. coli begins with hydrolysis of the enzyme beta-galactylase. Oh, that was definitely missaid. The gene encoding beta whatever. LAC-Z is part of a coordinately regulated operon containing other genes required for lactose utilization. Which following correctly depicts the interaction of the lac operon when lactose is not being utilized. So I don't have lactose. Let's think for a second. Okay. The point of lac operon is that I need to make the things to break down lactose. There is no lactose. This is an inducible operon, so it's going to have to be off and get turned on. So that means that A and C are not right, because these are both showing that they're on, right? These genes are on, okay? And I want to find something that's off and gets turned on. So then that would be B and D. These are the only ones that are off, right? Because I've got the repressor bound to my operator. Okay, so I can't have RNA polymerase binding because they're bound in the way. So I have to think to myself, lactose, it's not being utilized. So would I find lactose in the picture? Well, no, I wouldn't. So that shows me right here that I've got my operator blocking, okay, because it's an inducible operon, and I have no lactose, so lactose is not there. So I see that my operon is off. Since eukaryotes don't have operons, is there any specific we need to know? So in terms of regulation, they're just going to have a, sorry, let me read the question the person said. The person said, since eukaryotes don't have operons, is there anything specific we need to know about how eukaryotes regulate protein synthesis for the AP exam? So in terms of regulation, we're just talking about activators and inhibitors. So if there's an activator, it means that it's going to bind to activate and turn on that gene. It's going to act as a transcription factor to stimulate the RNA polymerase to bind to the promoter, and then of course transcribe that gene. Versus if we have like an inhibitors would of course inhibit the RNA polymerase from binding, which would then of course turn off that gene. So here we see 2017, number 6, we have a comet assay in a technique. So real quick, the reason I put this in here is because this is weird. This is different. This is unique. The first time I ever saw this was when I was grading when they posted the questions for that year in 2017. And I'd never heard of this before. And so don't get tripped up. If they give you something you've never heard about before, take a breath, take a breather, and look for the biology. Okay. You all know this information. You all know this content. Okay. They're not going to give you something you don't know. Okay. They may package it up in some funky, crazy way that's going to throw you through a loop, but you know the biology. So just look past the words you don't know and look at the biology that you do know and make sure that you're answering the questions that you know, because they're just asking you the biology. Okay. So common assay is a technique used to determine the amount of double-strand breaks in DNA or DNA damage in cells. The nucleus of an individual cell is placed on a microscope slide coated with an agarose gel. An electric current is applied to the gel that causes DNA to move, electrophoresis. We know electrophoresis. And the DNA is stained with fluorescent dye. When viewed using a microscope, undamaged DNA appears at a round shape, and the fragments of damaged DNA extend out from the head, the tail. The length of the tail corresponds to the amount of damage in the DNA. So here I see that I turn on the electrophoresis. I see that the head is at the negative end and the tail is at the positive end, okay? And so all I'm seeing is that if there's breaks, right, I have smaller fragments, my smaller fragments will move and separate from a larger fragment. That's all I see here. And that's all that we see with electrophoresis. There's nothing that we don't know at this point. We're good, okay? So explain the movement of DNA fragments, identify one property of DNA, and provide reason to support how the property contributes to the movement during the common assay. So what do I know about DNA? And why would the DNA be moving? So look at the picture. I see there's a negative end. I see a positive end. I know DNA is negatively charged. DNA is negatively charged because the phosphate in it is negatively charged. There we go. Negative charge, and is being drawn toward that positive end. Okay, that's simple. Another thing you talk about is the fact that we're looking at that there's these breaks in it, right? So a smaller fragment is going to move toward the positive end faster than a shorter, I'm sorry, I said that wrong. A shorter fragment will move towards that positive end faster than a longer fragment. So DNA can be different sizes, and different size fragments are going to move at different rates, okay? Again, you know the biology. Don't let them confuse you with an example that you've never seen before. That's okay. They want you to see, can you apply your science knowledge, okay? Which you do know. You can apply that science knowledge. You know this information, okay? So in part B, in a different experiment, cells are treated with a chemical mutagen that causes only nucleotide substitutions. So substitution isn't breaking anything, right? All we're doing is substituting one nucleotide for another nucleotide. This is, you know this, okay? Don't get confused. Predict the likely effects of the common assay for this treatment. Okay, so undamaged DNA will appear as a round shape, and fragments of damaged DNA will extend from the head and make a tail. Well, did I damage the DNA? Nope. All I did was change one nucleotide for another nucleotide. So I would expect there to just be a head. So head only, or a head with no tail. Or you could talk about that the tail will be shorter than one that had double-stranded breaks. Okay, so again, don't let them confuse you. So here I have another one. And I kind of use this one more because I wanted us to look at different experimental design components, and it did have to do with genes, so whatever. Common bedbug is a species of insect that is becoming increasingly resistant to insecticides. Bedbugs possess several genes suspected of contributing to resistance, including P450, ABC8, and CPS. To investigate the roles of these genes in the insecticide resistance, researchers deleted one or more of these genes in different strains of bedbugs, as indicated in Figure 1. And treated the strains with the insecticide beta whatever. Each strain was genetically identical except for the deleted gene and was equally fit in the absence of it. The percent survival of each strain following the treatment is shown in Figure 1. So here I have that figure, right? I can see the survival of them. I can see which ones they have, right? So if they have the gene or don't have the gene, there's a negative if they don't have the gene, and then there's a positive if they do have that gene. And so the first says, identify the control strain in this experiment. So my control strain is going to be the one that I'm going to be comparing all the treatments against. And so since I'm testing how these genes are being affected, right? So I'm going to test if I don't have those genes. That tells me that my wild type, my strain 1 is going to be the one that has all those genes, and I'm going to be comparing the treatment against that. And so that's going to be my control. Then it says to use the means and confidence intervals in Figure 1 to identify the claim that ABC8 is effective at providing resistance to it. Okay, so in order to be resistant to it, I need to find that there is a high percent of survival. Okay, so what am I going to compare 1 to that would allow me to say whether or not ABC8 is effective? So let's just look at ABC8. So here's ABC8. 1 has it, 2 has it, 3 and 5 do not have it, and then 4 does have it. Okay, but I need to have something that I can compare. So if I compare 1 and 3 together, that would allow me to see that if only ABC8 was missing, would I find that they're going to be resistant? And so you can see here, okay, that their means do not overlap, right? And then their error bars don't overlap. So the reason I really drew this out here was just so we can kind of compare it to see what happened. So every single time that we were missing it, so you can kind of see that both of these error bars are missing. So the error bars of strain 1 do not overlap with strain 3, or the mean survival of strain 3 falls outside the 95% confidence interval of strain 1. Strain 3 shows a significant difference from strain 1. So they needed you to look at the means and the confidence intervals. And so when you're saying the error bars do not overlap, be very specific about which ones you're talking about. You were talking about strains 1 and 3. Strains 1 and 3, the error bars do not overlap, and the mean is lower when we see our percent survival with 3 when it's missing the gene than when it does have the gene. Be very specific about which ones you're talking about. Part B, P450 encodes an enzyme that detoxifies insecticides. Okay, so P450 is going to detoxify the insecticide. ABC8 encodes a transporter protein that pumps insecticides out of the cells. So this means that the insecticide gets in, but ABC8 pumps it back out. So it just gets it back out of my cell. CPS is going to encode an external structural protein located in the exoskeleton that greatly reduces the absorption of insecticides. So this means that the insecticide is not going to get into my insect. Based on this information in the data in Figure 1, explain how a deletion of both P458, and ABC8 results in lower survival compared to the deletion of CPS alone. Okay, so 450 and ABC8, their jobs are going to detoxify and then also transport the insecticide back out. Okay, so it gets rid of it and it also eliminates it. Versus CPS is only going to stop you from getting it. So if I look at this where I don't have 450 and I don't have ABC8, I can see them significantly lower, right? And so we're going to see that when it's deleted and CPS is present, the bed bugs cannot detoxify or pump out the insecticide. And so there's a lower bed bug survival. Okay, so because of that. Then if I look here at 4, I can see that only CPS was deleted. And so I can see that, well, we're still seeing that okay, so let's set our mind wrapped around this. I'm inhibiting the amount that comes in, but if it does come in, I'm still able to pump it out and I'm able to get rid of it. And so you're gonna see that those are present, I deleted, and then of course they can detoxify and pump out the insecticide, so there's a higher chance of the survival. So you really kind of have to apply what you're seeing in your prompt to your question. So I hope that was helpful. I know that there's a lag between when y'all see things and when I say things, as well as there's a lag between when I cut the video, like it doesn't cut some stuff. So I do have a little moment where you can write your Q&A in. So if there's any questions you have, I'm more or less just killing time right now, trying to let you catch up and have a time to write your question. And then so I'm just writing you, don't forget, Instagram, AP Bio Penguins. I've been posting review. I have Unit 7 and Unit 8 to go. I have TikTok. Who really knows about that? We don't really need that right now. And of course, you already know about YouTube because we're here and you're currently watching this video, so you already know about YouTube. So do we need to know a lot about DNA methylation and the other types of regulation, or mostly just operons? You could also think about like the regulation. And so thinking about like, now we have methylation. The methylation is going to cause your DNA strand to get supercoiled, and so it causes the DNA to wrap tighter. Actually, wait, I lied. Methylation is going to inhibit the ability for RNA polymerase for binding to it, and it also kind of supercoils it. And then if we acetylate it, you're acetylating the tail on the histone, which allows it to unravel slightly. A lot of those you might see a little bit. Can you explain how eukaryotic cells regulate gene expression if they don't have operons? So it's just having to do with activators and inhibitors. Activators are going to bind to specific sites to allow for RNA polymerase to bind, versus inhibitors are going to block the binding of that RNA polymerase. It's going to make it so it's not going to be able to do it. How much do we need to know about operons? It's in the standards. There's really no way to know which questions they're going to put on the exam. What time is next week's Unit 7 review? Unit 7 review, I don't think is for another like 2 or 3 weeks. I think that because Unit 7 is a beast of a unit. It's got like 13 topics in it. So I don't think we have that one until like 2 weeks from now. So next week we're off, the following week we're off, and so it's the following week. So 3 weeks from now is when we'll do Unit 7's review. If you go to my Instagram, like the second pinned page shows you the calendar, and it'll show you like what days we're doing the review. But the review is usually at 3 o'clock on Saturdays. I was on spring break, which is why we had to do this one a little late. Explain transcription factors, how they relate to regulation. Transcription factor is just an inhibitor or an activator that's turning on and turning off a gene. It just binds to a location on the DNA that allows the RNA polymerase to come to the promoter. Like that's, I think we put a lot of emphasis on it and we freak out a little bit. Anytime I've ever seen it on the exam, there's been a picture that kind of shows it to you. So how many hours should you study each day up until the exam? So we're currently filming this on April 6th. And so I would be spending at this point, depending on how many APs you have, I would be spending like 30 minutes to 45 minutes during the month of April, and then I would kick it up to an hour when you got into May, because of the fact that you're going to need to start doing FRQ practices, and you're going to have to do multiple choice practices, and so those would be taking a little bit longer. So at this point, you're doing a little bit of content review. We have exactly 40 days to the exam starting today, so that means you could spend 5 days per unit to get through to the exam. You could also probably do 2 units a week to get yourself to the exam. So you kind of create a calendar yourself. If you go to Marco Learning, I've got a study guide posted up there for you, as well as I have different guides that as we get closer and closer to the exam, I keep posting different ones because we all hit that I'm going to study mode at different times. Unit 5 through 7 exam on Monday, which unit should you focus the most? That is a question for your teacher. I do not know what your teacher is going to focus mostly on. I would focus, that's a weird unit to have, 5 through 7. That's a lot. I don't know. Anyways, so are there any other questions we have about Unit 6, which is what this review today was on? So going once, going twice. Anyways, hope that today was helpful. Remember, AP Bio Penguins are just success. Have a wonderful day. Bye, y'all. Killing time, killing time.